and in the chat box, tell me when you think you're going to get vaccinated, or if you have been vaccinated. Go ahead and put in the box in the chat window when you hope you'll get vaccinated, or if you've already been vaccinated. Nice work, Z. You must be a healthcare worker. Oh, very nice. Yeah, hopefully late spring. I read this report saying maybe, I think Pfizer's, they're slightly behind, but maybe able to be on track. So maybe by the, I don't know, late May, they were saying. All right, 1205, welcome. I'm sorry, go ahead, Z.
Yeah, I said I teach. That's how it got it.
Oh, I see. Well, welcome everyone to our discussion today. We're going to talk about the Pfizer vaccine. There's some great molecular biology. In fact, an entire semester's worth of information is contained in this Pfizer vaccine. And my eternal regret is that I only have you for 25 minutes. I'm going to share my screen with you, and I'm going to show you a few slides, and then we'll get through to an activity. First, this is when you hear 4.284 kilobases, I want you to say it in the same voice that Doc Brown said his 2.21 gigawatts, because it is equally mind-blowing, especially knowing what each one of these bases does, which I hope to discuss in this presentation. Before we begin, it's important to remember the central dogma of biology. So you understand that there's DNA, and that DNA gets copied in the form of RNA, and then RNA is translated into protein. Proteins are the workhorses of the cell. We are who we are and function because of how we function, probably because of, some estimates put it between 20 and 100,000 different proteins. Mostly it's about 20 to 30,000 genes, but sometimes those genes get translated differently. There's alternate splicings and alternate interpretations and translations of those genes, but a lot of proteins, let's just say that. All that protein, the coding for that protein comes from our DNA. Now you remember DNA from high school biology. You know that there's adenine, guanine, cytosine, and thymine, and that RNA is adenine, guanine, cytosine. But instead of thymine, RNA has another base called uracil. And it's also denoted in that RNA is typically single-stranded, whereas DNA is typically found in a double-stranded helix. If you could take all the DNA bases that comprise a human, there are about 0.2 billion base pairs that make us human. And so I've just put here on this slide in really small print, just 100 base pairs of what we might think a typical gene looks like with G's, A's, T's, and C's. Before we get to a protein, it's important to understand where we've come from. Remember, there's a gene, and that gene we canonically think of as being responsible for a single protein. And going from gene to protein, there's an intermediate step of RNA. And that RNA needs to have special features if it's to be recognized by other enzymes and translated correctly. And the cell has evolved mechanisms and proteins that recognize non-self RNA, or RNA that comes from a virus or from a bacterium. And in differentiating its own RNA, there are molecular modifications, chemical modifications that happens to the mRNA to protect itself from degradation. Eukaryotic mRNA or RNA has introns. Remember, these introns must first be removed before translation. In addition to these, there are two steps that I like to summarize as protect the ends and splice the middle. So we have a 5' end. That's the end. As you're looking at the gene, that's the end that's on the left side, or the upstream region of a gene. The 3' end is the region that's downstream of the gene. Both the 5' and the 3' end have modifications that protect the RNA. And anything in between, those introns get spliced out. They are removed, and the exons are brought close together, and then translation occurs. Now, if we'd had sufficient time, my first activity is to walk you through the process of cutting out I gave you a 4-page worksheet, and the last page is a mRNA molecule that is yet incomplete. And so I was going to go through it with you. We were going to remove the introns. We were going to splice the exons. And then I was also going to have you use your marking pens to denote the chemical modifications that occur. But we'll just skip those steps, and I'll abbreviate them here by saying the introns are removed, and the 5' end is protected, and the 3' end is protected. Now that 5' end has a special cap, and it has a chemical modification to the first nucleotide that lets other enzymes recognize it as being RNA. Also, the 3' end has several adenines appended to it. You remember adenines as being one of the nucleotides that comprise RNA and DNA? Well, a series of adenines, sometimes 20 to 100 different adenine molecules, are appended to the 3' end. So now the cell, once the introns are removed and the cap is appended and the poly A tail is there, the enzymes transport it from the nucleus into the cytoplasm. And it's here in the cytoplasm where another enzyme called a ribosome recognizes the RNA and can begin translating it. So, it's just a micro world of molecular biology here in this cell. It's so interesting to know what happens to RNA going to protein. I spent a lot of years in a lab researching plants, and in particular, a plant that produced a special protein. And this special protein made plants resistant to a fungus infection. And there was a lot of unknowns about how this protein went to a special place within the plant cell. Well, all proteins, if you think about it, the cell is kind of like an Amazon warehouse. And we all love Prime Day and how those packages come 1 or 2 days after clicking buy. Well, the cell, much sooner than 1 or 2 days, needs to deliver proteins in the right spot and at the right time. And one way it does this is to recognize a molecular cue or an address that's in the RNA or the growing peptide, the protein. And it's like a zip code. And once the cell recognizes that zip code, it can go anywhere in the cell, maybe to the the membrane, maybe to the lysosome, maybe to the endoplasmic reticulum, maybe to the Golgi apparatus, maybe to the mitochondria. Proteins go to where they need to go because of molecular signals within them. So now that's kind of a brief background of what we needed to know in order to understand what Dr. Hackham says is the miracle of the Pfizer and other vaccines like it, an mRNA vaccine. I just want to bring to your attention 3 miraculous features of the Pfizer vaccine. First of all, it's an RNA vaccine. It used to be, you might have heard of Edward Jenner, how in the 18th century used cowpox to inoculate individuals from the smallpox. And it just so happened that even though cowpox virus and the smallpox virus, even though these are different viruses, they're similar enough that inoculation with the cowpox virus resulted in immunity from smallpox. And this required proteins from the cowpox being recognized by antibodies. And those antibodies then, when the smallpox virus came along, were able to neutralize the smallpox virus. This virus is special in that rather than inoculation with another virus. It is a virus that the cells haven't seen before. And instead of the physical presenting of a virus exogenously, that means coming in or coming out and within, it is code. Not just the protein, but the instructions to make that protein are uploaded into the cell. And it's never been done in this way before. Now, if you think of the vaccine as a code, then you can imagine a carrier for this code to be a liposome. A liposome is really just a small sphere that looks like and has some of the same features as the same sphere that has a human cell. I like to think of this sometimes as a flash drive. Remember back when we lived not so long ago, we could take a Word document or a PDF file, and then we could transfer it to a flash drive. And then that flash drive could be unplugged from one computer, inserted into another computer, and then uploaded to that computer or transferred to that computer. It's analogous to our mRNA vaccine. Instead of a protein being presented, the code for a protein is uploaded into the cell, and then the cell generates that protein, which is subsequently observed or seen or presented to antibodies. So it's interesting because it's not real. It is a vaccine in that it generates immunity, but it's really just a set of instructions. It's just 4,000 bases that generate a protein that generate the antibodies that comprise immunity. Another miraculous thing is that uracil, remember how I mentioned that A's, G's, C's, and U's comprise RNA? Well, uracil turns out to be slightly immunogenic. And for a decade-long quest has been to make RNA not as immunogenic, that is exogenous RNA that comes in. Early researchers noted that it would activate the immune response, and then therefore RNA coming in would be degraded. But we want, or the Pfizer vaccine wishes this RNA to be incorporated into the cells and to slip under the radar of detection. And so a chemical trick came about by replacing uracil with something like uracil called 1-methyl-3 prime pseudouridine. It's like uracil, but it's got a special appendage that functions as uracil, but slips unnoticed by the immune system. I want to say that I just want to show you a few images here of the GC cap. Well, the Pfizer vaccine also has a GC cap. It looks like this. And this doesn't mean much to you right now because you haven't had organic chemistry, but suffice to say this might one day be significant to you. And uracil being represented as 1-methyl-3 pseudouridine looks like this. So it's still uracil, or it has it doesn't hamper the translational machinery, but it escapes immune detection, which means it escapes degradation, which means it's presented in the cell and exists in the cell and therefore can be translated. So I'm going to show you a sequence, and if you were to replace all the U's with this pseudouridine, it would look something like this. But your worksheet doesn't have this. It just represents U. But you should know that it's not really uracil, but a modification of that uracil. All right. So now, if you have your sheet, I want you to open it up with me. So this is a handout that I gave you. It's 3 pages, and it's just G's, A's, C's, and U's. We're going to go through this base by base, and we're going to determine what every base in here does and its importance. So I'm sharing my screen with you. Also have your coloring pens ready. Okay, first thing, the first 2 bases, I want you to highlight. If you're digital or if you have a physical copy, go ahead and highlight those first 2 bases, and I want you to color them. You can choose the color. Those first 2 bases are the cap. This is the 5' end, the leftmost end, the upstream end of our RNA vaccine. And remember, Pfizer is trying to get this to look like just another RNA molecule, because once it's brought into the cell, once it's observed in the cytoplasm or detected in the cytoplasm by translational machinery, it can be translated just like a protein. And we'll see here in a little bit what that protein is. But right now, bases 1 to 2, I just want you to highlight the first two bases, the G and the A. That represents our cap. Next, select base 3 to 54. So there's 3, and I'm coming down. You'll notice that every line contains 50 bases. So if I go 51, 52, 53, 54, there it is. This is the 5', oh, that's a terrible color. This is the 5' untranslated region. Now this untranslated region is untranslated, which means it doesn't make up part of the protein, but it has implications for how long the mRNA resides in the cytoplasm. It has promoter elements which recruit the ribosome and other enzymes for translation. There's a lot of messages that can be decoded in this 5' untranslated region. Now, next, if we had a sufficiently long time, I would also share with you the amino acid code. If you remember from your molecular biology, you might have heard of the word codon. That means that every 3 bases, every 3 nucleotides code for a special amino acid. If the language of DNA and RNA is nucleotides, the language of proteins is amino acid. Remember, these are like glutamine and thymine, or glutamine and cytosine, and 18 other amino acids. Well, these 3 right here, this AUG, that's a special codon. This codon tells the ribosome when to begin to translate. In the amino acid code, this is ATG, but remember the T has been swapped out for U in RNA. So this tells the ribosome, I'm a protein, translate me now. And so this is where it begins its translation. So then it begins its translation right here, and we're going to go all the way from let's see where we're at right now. We're at base 54, so we want to code from 55 to 102. So we're going to go from 55 all the way to 102. Now, this is important because these end up being amino acids. And I mentioned earlier how amino acids, proteins need to know where to go. This is it. This is its zip code. This is a lot larger than 20850. I don't know what the zip code is over there in Beth, Tfiloh, but it's a lot more than 5 digits. This zip code recruits other proteins, and it recruits this protein to be delivered to the endoplasmic reticulum. Because this is the same pathway that the spike protein from SARS-CoV-2 takes. Once a virion integrates with the host cell and is brought in, its own spike protein is translated and then brought to the endoplasmic reticulum, shuttled to the Golgi apparatus where it becomes glycosylated. You might have heard of that as sugars kind of decorating the outside of this protein. And then from there, it gets presented on the membrane surface, or it gets repackaged actually in other virions. Well, here, this sequence recruits other machines that allow for the export of the spike protein to the cellular membrane surface. So once we have that, now I want you to select bases 103 to 3,879. This 103 to 3,879, which is 850, 860, 879. This is the money. This is the big dog. This is the spike protein. And if we had enough time, we would go to a database and we take the spike protein code, and then we take this spike protein code and we'd line it up, and you would see that it's mostly the same. There are a few changes here, and there are changes that need to happen because the spike protein also has changes. And when it interacts with the ACE2 inhibitor, ACE2 is a receptor in certain cells. And when the spike protein interacts with the ACE2 receptor, it undergoes a shift in its structure. And rather than have that shift, there are modifications in this spike protein that keep it open. And that's important because that means it's more open to being detected by antibodies. So really what happens is the lysosome binds with the cell. That liposome releases its cargo. That cargo is an mRNA. It begins to be translated. It gets translocated. It gets shipped to the endoplasmic reticulum. From there, it gets presented on a cell membrane surface. This occurs mostly in dendritic cells. They're kind of the shopkeepers, the sweepers, the cleaners of it's just a certain kind of cell that has a special job, and it mostly eats, or endocytosis, that means takes up these liposomal particles. And once once a mRNA is brought into a dendritic cell and spike protein is put on its surface, it's recognized by T cells. And T cells are kind of where the immunity begins. T cells can send other instructions to begin generating antibodies. And there's no danger here. There's no harm in presenting the spike protein on the surface of the cell because it's recognized by antibodies. And once antibodies learn to recognize the spike protein, if it's brought up again in a cell, say through the real spike or the SARS-CoV-2 virus, a lot of antibodies can be expressed that will neutralize the virus. Now, I know we're out of time. I just want to share with you the last couple of coding sequences. And that is, you have a 3' untranslated region here. So from here to about here, this is the region, again, that's not translated, but we kind of need this region to trick the cell into thinking that it's a spike protein. And then after that, this region, this is the polyadenylation tail. So this gives it also stability and lets the cell know that it's not foreign DNA, but is self DNA and therefore should be expressed. So in a nutshell, that's what happens when RNA is put in a particle and then that particle binds to a cell and the spike protein from the virus is presented on the cell, which is recognized by the antibodies. That's what 4.284 kilobases gets us. Questions?
I have a question, Dr. King. What was go back to like high school, college, what was the event in your life that really got you passionate in this area? What was the moment?
My first job out of university was working in a lab. We sequenced the BRCA1, BRCA2 genes. These are genes that predispose to breast cancer. And I remember designing a genetic test and just thinking, wow, I can use enzymes to amplify a particular region of DNA a quintillion fold. And then it can be sequenced. I can understand every G, A, T, and C in here and understand if there are mutations and what those mutations bring or might or cause within the protein. And just that linkage between DNA code and DNA protein really excited me.
That's good. And students, if you have questions, if you're too shy to ask, you can just like put it in the chat to me privately and I can ask Dr. King, or you can just put it in the chat yourself. When you're not donating your valuable time to Beth Tfiloh giving presentations, what are you researching right now?
Right now, I'm a senior scientist at a biotech company close to Baltimore called LuminUltra. We're making COVID kits, and I'm attempting to increase our throughput capacity. That means the number of samples that we can do by using robots. So right now, a lot of my day is spent programming. I like to think that I program cells, and I also program robots. And additionally, I also express proteins. I take I take a little bit of DNA and I give it to E. coli, which is a popular bacteria. And E. coli will express a protein that it's never seen before, that in fact belongs to a whole nother organism if it's just fed the right chemical signals. And so that's what I do. I express polymerases, luciferases. Those are proteins that glow. I express a lot of proteins, sometimes cancer-causing proteins like this, sometimes human proteins, sometimes jellyfish proteins. So it's a great time to be alive and to be active in the area of biotechnology.
And what are the, if someone's interested in biotechnology, what are sort of the courses in college that they really need to focus on and emphasize and concentrate in?
And you might say biology, and that's true. I have found my chemistry class being immensely important, understanding concentrations, acids, bases, titrations. If you learn chemistry, you can learn biology. Because all of life at the atomic levels is just a discussion of chemistry. And I've also found my math and physics courses equally important and exciting. It's what helps me, it's what originally attracted me to coding, and also to think of life as a set of rules and a set of instructions that cells and other organisms just follow.
Great. Any questions at all? All right,
I have a question.
Yep.
What are the specific, so is the red part that you highlighted, is that what codes for the spike protein? Is that what you said?
The yellow is the spike protein.
Oh, okay.
That's about 3,000 base pairs right there. So this is the same code that the virus uses, just with like a few slight modifications. The red represents the polyadenylated tail. So remember, Pfizer wants this to be as close as possible to the real deal, and the real deal has a tail. That tail has a lot of A's in the end. That's the tail.
Okay, and I was just wondering, what does how do these spike proteins that they made in, I guess, the vaccine, how does that help? What does it do once it gets into the cell to kind of destroy the virus?
You've probably all seen a picture from the CDC of a virion particle, this. Well, those red nodules at the end, those are the spike proteins. Now imagine instead of the gray is the virus protein that packages this all up. Instead of the gray, imagine it just being a cell. Imagine it being one of your own cells, which in a few weeks it will be once you receive the vaccine. The vaccine creates spike protein in your cells. Those spike proteins recruit antibodies. I have a picture of that might look like this. So if the spike protein is red, the blue antibodies recognize the spike protein, although they're being tricked because it's not real spike protein from a real virus. It's just the code that your cells have processed that mimics the spike protein. And so once the blue antibodies recognize the spike protein and generate immunity or generate a response, then the next time your body sees the actual SARS-CoV-2, it can generate enough antibodies to neutralize the virus.
Good question, sorry.
I have a quick question as well.
Yep, Jacob.
So Mr. King, have you ever done like any teaching before? Have you done any like freelance seminars like this before?
I love teaching. I teach the best course at the University of Maryland. It's called BSCI 414, Recombinant DNA Technology. And it's amazing because this semester we expressed the spike protein, but we did it in bacterial cells. You might have heard of the receptor binding domain. We also expressed that. We even generated our own SARS-CoV-2 test with our saliva. Good news, I'm COVID negative. It's the best. It's an intro to biotechnology, and it is delightful.
I was just wondering because if I had you as a teacher when I was coming into high school, I would probably be planning to major in biochemistry right now.
You're the best, Jacob.
Very
Thanks
good.
for that.
That's a very nice note to end on. Does anyone else have any questions? All right, Dr. King, thank you so much for your time. I know it's really valuable, and have a great day.
Appreciate it, friends. Have a good one.
Bye, guys.
Thank you so
you.
much.
Thank you. Love you.
Thank you very much, Dr. King.
Mr. Green, have a good one.
You too.